Hello, everyone. So I will be talking about the RM systems in Staphylococcus aureus and their role in the selection of phage production hosts. And I will go first very briefly through the staphylococcal phage resistance mechanisms and especially like RM systems. So the resistance mechanisms, they target mostly the adsorption, biosynthesis, and assembly stages of phage life cycle. And one of the main or major phage-resistant mechanisms are the restriction modification systems that consists of like three subunits, which are the specificity subunits that recognize a certain DNA sequence. Then there are the methylation subunits that methylate the DNA, and then the restriction subunit that restricts. And the topic of the talk is this small phage EBHT that is a lytic Rosenblum virus isolated from a pig snout sample against a clinical Staphylococcus aureus phage DSM 104437. And during the work, we have been producing the phage in two different hosts. So this original clinical isolate and then an alternative host 19A2 that is actually a pig isolate. And we used the we differentiate between the two phage variants produced in the host between the EBHD produced in the original host and MEBHD produced in the alternative host. And during other experiments, we noticed that after producing in the alternative host, the EBHD infection efficiency in the original host decreases quite drastically. And we could see that this change in the host range, it covers, it's quite drastic. So out of 162 clinical and pig isolates, the EBHD infected 89 strains, while the MEBHD infected only 32. So in the work, we wanted to find out what actually happens during the phage life cycle that leads to this host range change. So we had three options for why this happened, and the first one was that there is mutations in the genomic DNA. The other alternatives were that there is interruption in the phage adsorption due to post-translational mutations, or then the third alternative is that the genome replication is interrupted. So first we purified both phage variants and sent them for sequencing and compared the genomes, and they were 100% identical. So we moved on to the next hypothesis, whether there is interruption of the phage adsorption due to post-translational mutations. And we did a proteomics analysis for the purified phage particles. And we found out that the proteins that are encoded in the phage genome, the analysis identified all the same for both phage variants. Meanwhile, we found that the host originating proteins in the samples contained some differences, and we looked a bit closer at there are some peptidases originating from the host that were present in both phage variants, but they differed between depending on the production host. So we tested the phage adsorption if these host originating peptidases would affect the phage adsorption, and it was a no. So both phage, over 99% of both phage variants adsorbed on both host strains. So that meant that we moved on to our third hypothesis, and whether the phage genome replication was interrupted. So we did one-step growth curves, as shown in the A and B images. And we can see that actually the burst sizes are much higher in the alternative host 19A2 for both phage variants. And it's quite clear that there are no new phage particles produced in the new MEBHD particles produced in the original isolation host. And we can see a similar result when we studied the genome replication with qPCR assay. We could see that in the PEBHD genome was replicated immediately after infection in both host strains. Meanwhile, the MEBHD DNA is not replicated in the original host. So I took a bit closer look on the we sequenced both host strains, and we studied the phage resistance mechanisms in both host strains, and we found that there are different number of genes related to the RM systems in the hosts. That's the main message of this massive table. And to make the connection, if these the RM systems actually affect the methylation of the phage genome, and therefore the phage host range of the phage, we did we sequenced with PacBio method both phage variants, and we found out that they had both phage variants had unique methylation patterns. So the data shows that there were in total 23 methylation motifs found from both phage variants, and only one of them was shared between the PEBHD and MEBHD variants. And mostly the identified modifications were unspecified, but there were some M6A type of modifications. So in conclusion, we have a situation where, depending on the production host, we can see that the phage genome is methylated differently, and that defines if the phage infection can proceed. And so EBHD host range depends on the production host. And we can also see very clearly that both phage variants infect much more efficiently the alternative host, which also leads to a much narrower host range. And the PacBio data, we can show that the methylation patterns are different between the phage variants, and this is something that should be considered when we design the production of phages for treatment purposes. So finally, I want to thank all the collaboration and everybody who has participated in this work. So thank you.